What is up, Brad fans? How you doing? How you living? Today we're talking about ants, but maybe not in the way that you're expecting. And that's because my guest today, Yuko Ulrich, is not an entomologist. She's not an insect scientist. Rather, she is a behavioral ecologist and evolutionary biologist by training. And her work is really focused on social behavior, specifically the interplay between social organization and disease resistance in animal societies. So yes, there will be reference to the pandemic in this episode. So if your research interests center around social behavior, group living, and its relation to disease dynamics, you might think to jump to an organism like chimpanzees. They're the most related to us. They're the most similar to us. We've heard genetically and all of these things. We can kind of see in their living our reflections of our own. But if you want to ask very specific and fundamental research questions about social living and social behavior and how these things affect our success or our risk to disease, you're actually much better off studying the organism that has taken social living to the extreme. Ants. For one thing, ants are a little easier to bring into the lab and observe. But as we'll hear from Yuko, there's a very specific type of ant, the clonal raider ant, which is particularly well suited for asking these questions. And she'll explain all this in the conversation, but it has to do with controlling for confounding factors like age and genetics. This species of ant also doesn't have a queen, which again plays a role in how well suited it is to answering some of these fundamental questions in a controlled experimental way. And beyond these sort of technical experimental benefits, as we've mentioned, ants and their relatives in the group of eusocial insects have taken social living to the extreme. And they've been very successful. Ants, as Yuko tells us, have come to find a way to live in nearly every single ecological niche on the planet. And they're one of the largest sources of biomass. So just by weight, they're one of the largest organisms, groups of organisms on the planet. So it seems like a no-brainer then if you want to ask questions about disease dynamics in social groups or the benefits of living in a social group, why not look at one of the most successful social organisms on the planet? Now, at this point, you might be asking yourself, don't we already know the answer to these questions? Whether group living is successful or not? We just talked about how successful it is for ants, and we know how successful humans are. Or when it comes to disease dynamics, the question of, does your behavior, what you do in the group, where you go, what your role is, expose you to different pathogens? Your intuition is to probably say, yes, it does. Again, something that we may have experienced during the pandemic. Who we interact with, where we go, what we do for a job might expose us to different types of risks and pathogens. But this is where evolutionary biology and experimental biology becomes so important, because without actually asking and answering these fundamental questions, we can't go further and then ask the next level of question. What is it about group living that makes it so successful? Is it an efficiency thing, a scaling up thing, the division of labor? Does it allow us to specialize? Is that what provides the benefit? Can we maintain homeostasis better if we all have our little tasks that we're all trying to do? When it comes to diseases, yes, what you do, where you go, will affect your disease risk. But then, how does the group respond? What are the defenses that a group employs in order to protect individuals or the group itself from these types of risks? And this is where things get really interesting. As you'll hear, ants have developed some pretty fascinating ways to guard the group against infection. And this was actually a really important takeaway for me, because I think we sometimes forget the benefits of group living, especially in this polarizing, isolating time that we're living through. As a group, we actually have a lot of defenses available to us that solitary organisms don't have. And be sure to stick around to the end of the conversation, because whenever you're talking about group living individuals' behavior and their disease risk, how we respond to infections, we are going to bump up against that classic arms race between parasite and its host. And what happens when the parasite takes that to the extreme? That's right, host manipulation and zombie parasites. And now Yuko was cautious, and she made sure to point out that they can't yet prove that the organism, the parasite that she's using in her infection experiments with the ant, is a true zombie parasite. She and her lab group have found some tantalizing clues as to how this parasite may be influencing ant behavior for its own benefit. And she describes some pretty interesting and kind of funny experiments that they're now undertaking in her lab to start to tease this out. So I really want to thank, again, Yuko Ulrich for joining me on the show. This was a really fascinating conversation for me, given my background in parasites and ants. And so it was a real treat for me, and I hope for you too, to hear from someone who is so knowledgeable on these topics, who has such expertise and interesting perspectives, who's done such great work in this area. And I want to point out again that this chat was a wonderful insight into and reminder of the importance of experimental biology for answering these fundamental questions about traits that many organisms on the planet share, including us humans, and which again may seem obvious on the surface, but are a little more complex once you start moving to those next level questions. But of course, before we get to the conversation, I have to ask you to rate, review, subscribe to the podcast wherever you're getting podcasts. This is one of the biggest things you can do to help out the show and increase our visibility on these platforms in the algorithms. We also now are available on YouTube, podcasts for YouTube. And we'll see in the future if we expand into video. Let us know in the comments of our social media, Instagram @2bradforyou, X @2bradforyou, or wherever you're getting your podcasts, in the reviews, in the comments. Let us know if you'd like to see video, if you'd like to see some shorter clips, some dives on some topics on a YouTube channel. That would motivate me to maybe create that content for you. Anyway, rate, review, subscribe. Biggest thing you can do to help us out. Follow us on social media @2bradforyou. We are on X, although not very active there, and Instagram, where we tend to be a little more active. Either way, the handle's the same, @2bradforyou. You can always reach out to the show as well via email, 2bradforyou@gmail.com. I will read everything you send and try to respond either on the show or personally. Thank you so much for listening, everyone. And here is my fascinating conversation about evolution of social living, disease dynamics, and the clonal raider ant with Yuko Ulrich. Welcome, Yuko Ulrich. Hopefully I got that pronunciation somewhat correct. Thank you so much for being here on the show. I'm really excited to talk about ants, but not just ants, what ants can sort of teach us about different things that are applicable to human lifestyle. So group living, social living, and then how we deal with infections. So this is going to be really exciting for me. I used ants in my PhD work, so I'm a little familiar with the insect. But why don't you introduce yourself to the audience? Give us a little bit of where you are now, where you're working now, the focus of your research, and maybe you could explain to us how you got into this research and working with ants. Were you always interested in insects, or was it something along the way, your academic journey, that triggered, this is a system I could work with?
Yeah, thanks. Thanks so much for the invitation to have this conversation with you, Brad. I'm a so-called Lise Meitner group leader at the Max Planck Institute for Chemical Ecology. That's in Jena, Germany. And I've been here with my group for about 2 years and a half. I'm a behavioral ecologist / evolutionary biologist by training. And I've been working on all kinds of social insects ever since I was a master's student, so for a long time now. And so I kind of started working on social insects because they are so kind of incredible in evolutionary biology terms, right? They have evolved those really pretty unique forms of social organization. They show some of the most sophisticated forms of altruistic behavior and so on. So they really do things that no other animals do. And so for that reason, they take a really prominent place, I think, in evolutionary biology classes. And this is where I really fell in love with the topic a long time ago. And then I decided to do my PhD on social insects. So I was on bumblebees, and I studied disease already at that time, disease transmission in colonies of bumblebees. And then for my postdoc, I decided to switch system because I had heard of those strange ants that I didn't know existed before. Those are the clonal radar ants, and that is still the system I work on now with my group, and that we will be, I think, talking about.
Yeah, exactly. Yeah, it's always fascinating to me because the social insects, everyone's familiar with the bumblebees, the wasps, the ants, termites, right? We all see these things, but they seem so foreign to humans, right? Like the hive lifestyle, the queen, they're all sisters. It's a very seemingly different lifestyle than humans. But as we're going to discuss today, there's a lot of sort of similarities when it comes down to, social living, organization, all of these kind of things. So I'm just fascinated by this topic. I'm trying to think of the best way to approach all of the things that I want to talk to you about. So maybe we can start with, well, you could maybe advise me. Does it make sense to introduce the ant, the species first, and then go into sort of the evolution of social living? Or do you want to talk about the Nature paper that you did, the evolution of social living, and we'll introduce the ants along the way? Kind of put you on the spot there.
I think we need to talk about. I think we need to talk about the ants first, because the study really wouldn't have been the same, or might not have been possible in that way without us even knowing that those ants existed.
Let's introduce us to the clonal radar ants.
All right. So I think most people will be familiar with the idea that social insect colonies usually have queens that are, the only reproducing individual or individuals in a colony, as well as workers who are less reproductive or often completely sterile individuals and who take care of everything else in the colony. They clean, they defend the nest, they take care of the brood, and so on. And that makes social insects quite interesting because they have evolved this really sophisticated form of division of labor, right? Like the reproductive division of labor. And you also find other forms of division of labor between the workers who take on, for example, different tasks. But those differences are often fixed at the adult stage. So a queen ecloses as an adult, as a queen, and then there's no way back, right? In most cases. And so that means that there's really kind of limited flexibility in those behaviors. And so around the time that I finished my PhD, I heard about this ant species, the clonal radar ant, which has a number of characteristics that makes it both super interesting and special, and also extremely convenient, in my view. And the first one is that they actually have no queens. So I know that might sound a little bit counterintuitive, and that's kind of not the norm among social insects, but that is super convenient because they, in most other social insects, a functional colony needs to have a queen, and then often that complicates experiments a lot, right? You need many of those queens. They are by nature genetically different from each other. You don't necessarily know their history. And then the colonies are those very large, complex units that you can't really break down. You often don't know what's in the colony. But in this system, because the clonal radar has no queens, you can make those small colonies that are just composed of workers. And each of those units is, completely identical to each. Those units are identical to each other, and they are fully functional without a queen. So you can take a large colony, you split it up in many smaller colonies, and all those colonies are good to go. So that's very practical. And yet they are able to develop. They still have kind of morphological differences that are small, but there, and which kind of vary along this worker-queen axis, right? So some individuals are a bit larger and they have more higher overreactivity and therefore higher reproductive potential. So you still have a little bit of variation along the worker-queen axis, but you have a handle on it.
Mm-hmm.
And that's quite convenient. So that's the first thing. They are queenless. And so colonies are made up of workers, and all those workers can do everything. They reproduce, they can, switch tasks and so on. They forage, they take care of the brood. And the other thing is that all those workers are also reproduce asexually. So actually the system has almost completely lost males as well. And the workers are females, as in other social insects. And they basically reproduce by laying unfertilized eggs that produce genetically identical copies of themselves. So within a colony, all the ants are genetically nearly identical. And once a month, usually, they will produce new ants that are, again, genetically identical to themselves. And that's really neat because that's also not super common, I would say, across ants. It happens, but it's not the norm by far. And that's really convenient because it gives you a handle and a control over genetic background, which is something that, in most experiments will often have an influence on pretty much anything you study, right? It can influence behavior, it can influence disease susceptibility. So it's really nice to be able to control that in the lab. And the final thing is that they not only, so those workers in clonal raider ant colonies, they not only reproduce asexually, so clonally, but they also reproduce in synchrony. And so in practice, that means that in any given colony of clonal raider ants, all the workers will lay eggs within 24 hours of each other, so within a really restricted time frame in synchrony. And they do this about once a month, about every 5 weeks. That's really peculiar as well. But one consequence of that is that all adults will emerge in really discrete age cohorts. So you have, ants that are kind of 1 month apart, and you have those very kind of neatly arranged age cohorts. And that is, again, super convenient because it makes it pretty easy to control age, to get a lot of individuals of exactly the same age. And that's important in social insects because age really also influences behavior, as well as many other traits. So being able to control all those features is kind of an experimentalist's dream to some extent.
Right. And that's I wanted them thinking here of trying to summarize this all. And I think that's actually, I'm glad we started this way because that's super important. That's one of the things when I was reading your papers that you mentioned, and it makes a lot of sense that if you want to sort of study anything experimentally using, animal models or otherwise, age, genetic background are things that are very difficult to control for, but they will influence a lot of the different traits that you're looking at, right? And so in this system, you actually have, because you can split these into smaller groups and they'll function fine as little colonies or little groups. You can have, you can actually make a bunch of replicate sort of groups that are the same age, the same genetic background, all of these things, and then run sort of experiments on behavior or whatever else it might be. And you kind of are controlling for these other confounding factors that you wouldn't be able to do if you were just observing animals in the wild, or say, even people that run experiments with mice or other animals. You don't have that level of control over these two really big factors, right?
Exactly. I mean, the closest thing are indeed those model systems, right? Where people have created inbred lines to control genetic background and so on. But that wasn't really available for social insects. And so with social insects, I think for most of my PhD, you had to deal with those kind of uncontrolled levels of complexity and diversity, which are super interesting, but, kind of can add factors in your experiments that you don't know about or that you don't really want to be there.
Exactly. Okay, so now that we see the system, we see the benefits of the system, we've been introduced to this really unique ant, social insect. The first thing that I'd like to then move to is the social living, the evolution of social living, which is something you discussed in a Nature paper in 2018. And it's, again, it's something that I think a lot of people, we realize that we're social animals. we understand this. You hear it in popular science stuff all the time. Human cooperation, that's been our successful trait. But we often make the comparison to chimpanzees or great apes or other social living mammals. But if we wanted to look at the evolution of social living, group living, it might make sense to look at something a little more, I don't know if ancient is the right word, but social insects could give us a good understanding of how that dynamic evolved and what are the benefits and trade-offs to evolving as a group living species. Am I on the right trail here?
Yeah, I would say so. Yeah, social living, of course, as human, we think of it as, we are the most sophisticated social creatures, right? We like to, that's kind of a natural thing to think because we have pretty complex social life, right? And I think some traits are uniquely human. But when you think of the animal kingdom, widely, I would say many people agree actually that social insects have pushed it arguably a little bit further than us in many ways, right? And what's quite interesting there is that they are extremely successful as well, social insects as a group, right? I think the numbers are they represent only 2% of insect species, but they account for over 50% of the total insect biomass. So they are really super dominant, and they kind of really, in just sheer biomass, they dominate many ecosystems, especially in the tropics. Of course, they are very small, so we don't really know. You can live a whole life without really paying attention to them, but they are there and they do extremely well. So, so in that sense, I would say there are many independent evolutions of social life, and they, of course, in our groups, so in primates and so on. But social insects have done it over and over again, and also extremely successfully, but in a slightly different way than we have, right? So, like, for example, they have evolved this reproductive division of labor that we don't have, but in many ways, then kind of. When you look at social insect colonies in terms of like their size, for example, and their success, they are comparable to human societies, I think.
Yeah. So what were the questions then that you were looking at? Because it seemed, if I understood the paper correctly going through it, there was a question of what would be the initial payoff, or there might be an initial hurdle to overcome in terms of benefit that an organism would get from social living that maybe evolutionary theory wouldn't really predict. I'm trying to think of a better way maybe to explain that for the audience. But the benefits of social living might not be obvious. So it's maybe peculiar that it was selected for in the early stages. Is that sort of the idea?
Yeah, absolutely. I think yeah, that's it. And so essentially, what's impressive about social insects is, those really elaborate, super complex forms, like colonies that contain millions of individuals, those super large queens and so on. But that's a pretty derived traits evolutionarily. And so things must have started in smaller groups and before these very elaborate forms of division of labor, right? And so really what we set out to do is to use the clonal raider ant, which doesn't have this, crazy morphological specialization, which doesn't have queens, which all individuals look more or less the same. We know they have the ability everybody can do everything in those groups. And so what we set out to do is to ask whether division of labor could arise in those groups, and also at which group size it could emerge, right? So because initially, of course, social groups were not composed of millions of individuals. They had to start small and they had to start simple. So what we wanted to see was whether, very simply, it could emerge almost spontaneously in very small groups of individuals that are near identical in that case. And so, and we thought the clonal raider ant, well, it's one of the few systems that actually allows you to do that, because you can make groups of slightly different size very easily. And you can make sure that group size is the only thing that varies, right? It's not like the bigger groups have more genetic diversity, or it's not that the bigger groups have more variation in age, because this could explain, more division of labor through other mechanisms that simply coming together and forming those small groups that becomes slightly larger over time.
Ah, okay. So the real testing variable then is group size, initial group size.
That was the focus. We wanted to see, and we measured so what we call fitness. We measured how well those groups did, because we wanted to also be able to have a readout. So we varied group size, and we quantify how that affected division of labor and also performance, right? And we really saw that those things came in hand.
Okay. So what was the performance metric? How do you measure the performance of a small ant group?
Yeah. So as in biology in general, success is how many genes you pass on. So it's like reproduction and survival. So those are the two things that we measured, and then you can combine them to in some kind of colony growth rate, which measures how well an individual does on average in a small group versus a large group. And we could see that really both. So those benefits of increasing group size kicked in extremely early, actually, earlier than we thought. And so we showed that groups that contained really as little as 6 ants had division of labor and also did much better than colonies of like 2 ants or 1 ant.
Wow. Okay. So there was a benefit right away, even at 6 ants, to you're going to have a higher fitness. They're going to start saying,
Exactly.
you're going to do this job, we're going to do this job. So does it say something about, is the benefit then, because this is, I think, the other question, is it division of labor? Is it that they can do things more efficiently? Are they, is there, like, what is it about then this group living that is so, that makes it more successful?
Yeah, that's really the core question, right? So when group living arises, you almost always see some form of division of labor. It's not only true for group living, but it's also true for, the evolution of multicellularity, for example, which is also an example where small units get together and form a larger entity, really, that is functionally integrated. And so the idea is really division of labor is at the core of those elaborations of life, right, into more complex units, and that this is the true evolutionary advantage, that it provides an advantage that is so large that it promotes, in this case, the evolution of social life. But how exactly that happens is still kind of an open question. And so I think in that 2018 study, we could really show that those things came together, and we propose an idea like that comes from our own data, for example, but we couldn't really causally demonstrate that increasing division of labor causally improves fitness.
Mm-hmm.
And that's because, we manipulated group size, not the behavior directly. But that's a kind of experimentalist, maybe nitpicking here.
Yeah.
But so what you see essentially is that, so the advantages, the benefits of division of labor could come about via different mechanisms, at least, in principle. One is that if you're a specialist, you can become better at what you do, either through learning or experience and so on. It's also possible that even without being better at a job, the simple fact that you don't waste time switching between tasks improves efficiency at the level of the group, right? You don't have to constantly move from one job to the other, and you don't waste time moving between those tasks. And what we observed actually also in our own ant colonies in that experiment is that there was an advantage in the sense that as soon as you reach a certain group size, the likelihood that one of the jobs that you have to do is neglected, is abandoned, like no one does it, that decreases significantly. So in our case, we looked at, for example, the likelihood that the larvae were unattended for. Mm-hmm. And I think that can be quite intuitive for parents, for example, that if you leave your young unattended for a while, they might fight with each other. So like those ant larvae, for example, are prone to eating each other even. And so we found that as groups became larger, and for the same amount of work per individual, the likelihood that the larvae were not attended for decreased. And so it could be that simply in larger groups, you become better at, performing all the tasks at a sufficient level all the time, rather than having those kind of large fluctuations. It's hard for a single individual, for example. You simply cannot do all the tasks at the same time, so you have to switch by definition. And as soon as you have two individuals, maybe you can, one stays home with the larvae, the other one goes out to forage and so on. And those effects seem to really scale up as group size increases.
Yeah. The other thing that stood out to me was the idea of homeostasis. So keeping everything,
Exactly.
Is that kind of what you're talking about here, or is that sort of another element to it?
Exactly. Yes. Yeah. So exactly. So homeostasis is this notion that you can maintain some parameter within an acceptable, within a good range. And the idea is that our colonies seem to be more homeostatic, so better able to maintain stable task performance, right? That they wouldn't abandon a task for any given time when those colonies were of a sufficient size. And the example I gave of the larvae would be homeostasis in, for example, brood care, right? In how well you take care of the next generation.
Yeah, so it's just when you have that division of labor, everyone sort of kind of specializing to a certain job, no job is being left unattended, every job sort of stays within that range of care or attention that it needs. So the homeostasis across the group is maintained.
Exactly.
Yeah, that's really interesting. I'm not super familiar with it, but there was a book called The Tinkerer's Apprentice, and I know it had some criticism from evolutionary biologists. I'm not sure if you're aware of this book, but it was based on homeostasis, the idea that groups and cells and everything are kind of striving for homeostasis, and that's one of the things that keeps these groups together, or keeps these systems functioning. And I've always, the idea of homeostasis and trying to, organisms sort of pushing against entropy to sort of maintain these bubbles of acceptable living conditions or acceptable ranges has always been a fascinating one for me. And that's kind of what poked out to me in this is that, that's one of the advantages of group living, is that you can really work together to keep everything within these, acceptable ranges.
Exactly. Yeah, exactly. And I think that's true at, all levels of biological organization, right? I think we are maybe more familiar with the concept of homeostasis in a multicellular body, but of course so a body has to maintain its temperature, has to maintain its core functions like, I don't know, digestions or osmosis and so on. And then the cells within the body also have to do that. And then kind of the notion that social groups also have to maintain the homeostasis is quite interesting to me, right? That those things are true at many different scales.
Yeah, it's that interesting sort of, and I don't know how much we can really derive from it. Maybe it's just, maybe not a coincidence, but the fact that it does kind of scale up from a cell to a group of cells to tissues to an organism to multiple organisms, it is a really nice, sort of scaling up that you can see in these things. And that's just, I don't know, in a way it kind of gives you a nice, a lot of times in science, there's not a lot of closure, it is always, but it's to see this sort of pathway kind of emerge. It's okay, that feels right. And I know that's totally unscientific. That's just my own personal enjoyment with the subject and stuff. But anyway.
Yeah. So in social insects, actually, this is an analogy. So there's this analogy that you might have heard of, the superorganism. That insect societies are more than just a social group. Actually, they are integrated to the point that you could consider the colony, first of all, as the unit under selection, that the evolutionary unit, but also as maybe a single organism, right? Because the queen is analogous to the germline of, a multicellular animal. The workers are the somatic cells, the liver cell, the neurons, and so on. And I know many analogies tend to have limits, and sometimes there's this tendency to try to make too much out of them. But this one analogy of the superorganism is one that I still like and still use conceptually in how I design experiments and how we do our work in my group, almost a decade on. So, this has been one that is kind of still has a lot of appeal and I find quite useful conceptually to use.
Yeah. That's always, it's nice when you have those, metaphors, analogies, that kind of thing that sort of maintain, like you said, there's no perfect one. It's just like model systems, right? Like there's no perfect one, but this seems to work pretty well. And so keep going with it.
Yeah.
All right. The next thing then, and this is what, really fascinated me about your work when my wife came back from the conference where she met you. She was like, you need to talk about the infection work that you've done with ants, because that was sort of my PhD was all about parasites, parasitism, ecology of parasites. So I've long been fascinated with infectious disease, epidemiology, all of these kind of parameters. And unfortunately, we all got introduced to that a couple years ago. So we're all now kind of familiar with some of these, terms, some of these, concepts. And group living is one of the things. We just talked about how efficient it is, how it makes sense evolutionarily, that it would evolve even at small group sizes, some of the benefits. But one of the costs is infection risk. So when one individual gets infected, they can then spread it to the group. And there's been constantly this, people call it the arms race, right, between parasites or viruses, bacteria, pathogens, and groups and individuals and all this stuff. So you looked at some of these factors and you were specifically interested in behavior. So where an individual chooses to move within its group, or is destined to move. I guess destined is the wrong word, but what their job is, dictates where they move in a group. And pathogens also have, particular places where transmission is higher. So there's a lot of factors going on here. So maybe I'll give it to you to try and explain your study rather than sort of throw out a bunch of stuff.
No. Okay, but you got it right. It's like a very good intro for the study. Yeah, so basically, I think the starting point for this work was that, and first of all, I want to say this work and the kind of proposal that went to it was done before the pandemic, right? Yeah. So this was not something that was actually triggered by the COVID pandemic, but that suddenly became, some of the questions became suddenly things that were on everybody's mind. So it has become surprisingly very easy to introduce that work, because you find that most people in the audience actually have become unwilling experts of, disease transmission, and then have had to think about those questions a lot. And so I think this would be quite intuitive for most listeners. So essentially, when you think of, as you said, social groups are very susceptible to disease transmission because of those high rates of interaction between individuals. So this is a major risk of group living. And in many social groups, there are many factors that will influence your likelihood to become infected by something, right? There's your genetic background, there's your age, there's where you go, with whom you interact, and so on. Many different factors that complicate the question, and often in like, for example, natural populations of animals, it's very hard to know which, to what extent each of those factors played a role in that individual animal being sick now, right? And we thought that, well, the clonal variant is quite nice again to disentangle those effects. And by then we had developed those tools that allow to really follow the behavior of each individual in a group, which we could deploy to address questions about disease transmission. And so the question that we wanted to address first here was, well, we've all been thinking a lot about, or like we are all well aware now that your own behavior is an important factor in, or like is going to influence your likelihood to become infected, because again, where you go, with whom you interact, what you do are all things that very intuitively, should affect the likelihood that you contract an infectious disease. But often those things will correlate with other things. So for example, in the context of the pandemic, we know that young people, of course, they are younger, but they also behave differently, right? So they might be more prone to go to parties, or they might, but young age also impacts the immune system. So, there might be different effects playing out in the outcome of an infection, and many of those are confounded, or like they correlate in a way and they're hard to disentangle. And so what we set out to do was to use the clonal reader ants. By that point, we knew that we would have differences in behavior, that we would have division of labor, even among clonal individuals of the same age. And we knew this from the 2018 study. We knew that those differences would emerge in small clonal reader ant colonies. And we wanted to know whether then those individuals that took on different jobs in the colony, that took on different tasks, were then at different risk of becoming infected, in that case with a nematode that we study in the lab. And it was the case, and it was very clearly the case. So the differences were pretty strong. So if you're a forager in an ant colony, and forager means you go out of the nests, you tend to explore, to look for food, and to cover a broader, also spatial range. Like you go to many different places. And we found that foragers were at a much higher risk to become infected. They become infected earlier, more of them become infected, and also they end up with higher parasite loads than their genetically identical nestmates who tended to stay in the nest more. So that's a very simple finding, but one that really shows that behavior itself, so behavior alone, can drive the risk of becoming infected. And also a direct consequence of that is that so the distribution of parasites in a social group really reflects division of labor within that group, right? To the point that you may, in those experiments, we didn't try, but we might have been able to guess what behavioral role an individual was in charge, or what behavioral role an individual had based on its infection load.
Yeah, and I think that's something that's, again, it feels intuitive to us, and I think especially now, like you said, this work was started before the pandemic, but now we've all become sort of accustomed to thinking about this, or we were all introduced to it. And that's kind of intuitive, right? Like it makes sense. If you're going to go out into the world where the parasites are, where the pathogens are, you're more likely to get more parasites, right? But it's something, again, that we don't necessarily think of on a deeper level other than that, or at least we didn't. Maybe now we do. But that your behavior and, in this case, like the job that you're required to do in the group is going to put you at a higher risk. But so then what is sort of the consequences of that, from an evolutionary perspective or from a group living perspective? Is the idea that, well, the group then has to, sort of devise ways to, halt the spread, or maybe it's in other groups where genetics does play a role. Certain individuals will have a different genetic disposition against different parasites. Like, what's the consequence of this? Now that we know, like you said, a sort of neat, simple finding that some people are going to have more risk than others. What's the next question? What's the consequence of this for the group?
Oh, yes. Okay. And first of all, I should be clear that I cannot give any public health
advice.
No,
for sure. Yeah,
so I don't know what we should do in human societies. I can tell you a little bit about how ants deal with those things, both at kind of the evolutionary level, what kind of behaviors they have evolved to deal with those things, and also at the proximate level, which is how they immediately react. So in general, in ants, normal ant colonies, in which you have normal demographic structure, like variation and so on, like larger natural colonies that you might find outside your house, it's always the old individuals that go out Or like there's a very strong tendency for older individuals to go out and forage and to be exposed to the external environment. And this is really one of the few things that seem to be true across the board. It happens in honeybees, it happens in ants, and I think virtually any social insects where this pattern, where this question has been looked at. And the idea is that, well, those tasks are riskier. They are not only riskier in terms of pathogens, but also predation and so on.
Right.
You're more like it's more dangerous to go out. And so there's an idea that basically then you give those tasks to the older individuals in the colony because those have lower residual value, lower remaining value to the colony.
Yeah. It's a very harsh calculation, but
yeah. I mean, nature is harsh, and so that's the idea, right? That's and then you don't expose your younger workforce to those risks, or you only expose them to those risks later on when they have already kind of contributed to the colony's success. So that's kind of one of the evolutionary explanations for how you can kind of mitigate that risk by sending a subpart of the social group to the external environment. And luckily we don't do this. Yes. And then also, so basically the idea is also that social insects have evolved ways to organize their colonies in a way that then limits the spread of those pathogens within the colony. So again, within a normal ant colony, not all individuals have the same value to the colony. Right? The queen is super important. If there's a queen, then she's super important because she is the only one who can reproduce. If the queen dies in many ant colonies, the colony dies. And so it's thought that social insects have evolved many strategies to kind of limit the spread of infectious disease in the colony. And one of them is that they seem to have restricted interactions between groups of workers who have different jobs, essentially, right? So that the foragers, who are the ones who go outside and are most likely to bring back pathogens to the nest, those foragers will not interact with the queen. And they will only have limited interactions with the workers that are the entourage of the queen, if you want.
Right.
And so there's a notion that basically the structure of social networks within a colony and the spatial segregation and the way they organize themselves has evolved to some extent, at least, to protect the colony against, uncontrolled pathogen spread. And so, and those structures of social networks, some people or other, not us, but other studies have shown that they can even change plastically upon exposure to pathogens, right? And that's really interesting. So they seem to be able to, they have those constitutive levels of defense, which is represented by how they are organized, at baseline in normal conditions. And when they detect the presence of a pathogen, they seem to be able to strengthen those barriers even. But what we saw in our own study was something different altogether. And what we observed is that upon exposure to those nematodes, so the individuals that are exposed actually tend to spend more time in the nest. And this, from the host perspective, from the ant's perspective, is not very good, right? So if you're infected and then you run back to the nest and then spend all your time, in the nest, that seems counterintuitive, because then you should be at a higher risk of propagating the disease.
Yeah, it's like the opposite of quarantine, right? Yeah.
Exactly. And so this kind of surprised us, to be honest. We were expecting the ants to do something different entirely, and we are still not sure. But this made us think that maybe the parasite itself, that the behavioral changes in the hosts, as we observe them, if anything, should benefit the parasite, which again raises a host of other interesting questions, some of which you have studied yourself, right?
Yeah. The idea of the parasite manipulating the host for its own benefit. Yeah. I think this is, again, like we would think, yeah, and obviously, yeah, you can't give public health advice, and it's a model system of ants is very different than humans and stuff. But it's fascinating to me, this idea that our relationship with pathogens, all of organisms' relationships with pathogens really does structure or influence evolution in a way, how we evolve, how we live, right? So the ants have evolved these, kind of barriers in their division of labor that is a protection against disease. Like there's so many things, other social groups, whether it's grooming or, things like this, like there's all of these intrinsic behaviors that are because of our relationship with pathogens in the outside world and stuff. And so in a way, again, I don't want to keep hammering on the pandemic experience, but that was a very shocking thing for most people. But when you think about it, it's really like, it's such an intrinsic part of life, of being a living thing, is dealing with infection and how that influences our behaviors and how our behaviors influences infection risk. So these are all like really sort of fundamental questions and really kind of fascinating. I wonder what you think of that sort of statement.
Yeah, absolutely. I mean, I always really try hard not to anthropomorphize my ants and, try to be careful not to extrapolate too much. But in this context, it was really hard not to draw parallels sometimes, right? Because, for example, this different exposure to disease that different members of a group are facing, we saw it in the pandemic to some extent in the context of, healthcare workers. They were in the front lines and they were taking risks, right? Consciously to be exposed to this pathogen in order to take care of other people, right? And ants do this as well, right? They do care for each other. So like you will often see them grooming each other when a nestmate is exposed to a disease. Instead of ignoring that individual altogether or just letting it to deal with the infection on their own. So there's a lot of helping behavior, there's a lot of care behavior that you see in societies that we also have, obviously.
Yeah, I think it's like, and again, not to like get too, draw too many conclusions or anything like this, but again, like a sort of big takeaway for me is kind of just that, like, maybe the response that we had to, maybe facing this sort of these kind of fears, in a large scale way for the first time, it seemed like so chaotic, right? When really you would think that like every person has sort of an understanding of infection. We all know to cover our mouths, like we understand the basics of it, and we understand that there's this sort of big groups are going to transmit things. And obviously we're not going to go as far as the ants to like sacrifice the old people, like that sort of thing. But I would have thought there would have been a little bit more coordination, and maybe it's just our group sizes are too big. And obviously there's a lot of other social factors and stuff that go into it. But it is really fascinating to me how we all sort of face this question. And we can now kind of look back and say, well, that made sense, that didn't make sense, that sort of thing. But again, to me, fundamentally, the principles they're really not hard to understand. And when you look at the ants and how they do it, it's really quite on display, right? Like you keep people kind of segregated as much as you can, limit contact between different, it's all very basic stuff, but it's really cool to see it and actually, test it experimentally and say, okay, well, we ran the experiment and, this is what happened, whereas you can't really do that in natural populations, as you said. So to move away from the pandemic, I'm sure people are tired of, talking about this too much, but we can't avoid it in this topic. I am curious then, you touched on just briefly there that the ants infected with this parasite did the opposite of what you expected. And that is that they spent more time in the nest. Now, it's very challenging to know whether that is parasite manipulation.
Absolutely.
Versus just a side effect of being sick, right? Like
Exactly.
maybe they have this infection, they feel, as much as an ant can feel under the weather. Mm-hmm. And then therefore it can't do its normal job, or it's somehow its normal behaviors are disrupted. But this is a very interesting area, right? So how much of it is parasite? How much of it is host? How much of it is coincidence? Is there a mechanism there? These are the questions that really, really fascinated me. And I think that it's one of these things that we see again in the popular media, right? Like the zombie parasite. And I think that sometimes it gets a little, a little overblown, in terms of, is the parasite really, my example is always toxoplasma. Is it really doing all of these things that people say it's doing, crazy cat one? So just maybe your thoughts on this dynamic, this host parasite manipulation, how it makes sense that it would evolve from a parasite point of view. And I know you can't maybe say in your system what's going on, but you did identify a few things in terms of where the parasite was infecting that might, show that there is a behavioral change, or there's a reason for the behavioral change.
Yeah. So again, the bar to demonstrate parasite manipulation of host behavior is very high, and I don't think it's easy to clear that bar, and we definitely haven't yet, although we would like to. And what kind of piqued our interest? So first of all, as you said, we were expecting, the ants to react by, isolating. Maybe the infected ones would stay away from the nest or whatnot. And we saw, if anything, the opposite, which came as a surprise to us. But then, of course, ants are known both for their super elaborate defenses, collective behavioral defenses against disease, but they're also known for some of the most iconic examples of parasites manipulating host behavior, one of which you worked on, Dicrocelium. And of course, we were aware of both. And so basically in the case, so as I said, we were working on parasitic nematodes. There's very little known on those. Actually, we had to really dig in very, very old literature to find anything that kind of any paper that would say something about nematodes infecting ant heads. But it just so happens that they infect a very social organ in ants. So they infect the head. We initially thought they infected the brain, and then it turns out that we were wrong. And the head is pretty small. It's easy to mix up organs, but they don't infect the brain, but they infect an organ that's just next to the brain. And that's a gland that is considered to be a very social organ in ants because it is involved in the storage and the social exchange of molecules that in turn allow the ants to tell who is from their colony and who is an outsider. And so basically, the simple fact that the nematodes infected that gland, and so as far as we can tell, that gland only, suggested that it should do something to social behavior, right? And so in the paper we published, we could just show that it did something to individual behavior, in the sense that in colonies where everybody is infected and of the same genotype, then you see this change in behavior. But the fact that we find it in that gland in particular raised the question of what it does to what we call nestmate recognition. So in other words, the ability of ants to tell who is from their colony and who is not. And so the experiments that we are doing now is to see whether it essentially affects the ability of ants to detect nestmates. And usually the way you run those experiments is that you put ants from different colonies in the same arena. And in normal conditions, if they are from different colonies, they will fight. So it sounds like an experiment that many, children have done in in their garden. Yeah. Well, we are doing it for real, and this time we use uninfected ants and infected ants, and we want to see whether there's a change in how they attack or accept each other as a consequence of infection, essentially. And this if we see effects there, this might in turn have effects on colony composition, colony structure, and so on. And so in terms of the manipulation itself, we do not know yet. And I think we would need to do a lot more experiments to conclusively demonstrate that there is indeed some, for example, neurological effect of the nematodes on the hosts.
Yeah. And again, just thinking of sort of a broad, like a big picture kind of, thoughts about this sort of work, it again highlights, even if it's not direct, parasite driven manipulation, it just shows how an infection can have consequences on an individual's behavior, which then has consequences on their group behavior and group infection risk and all of these things. So it's just, again, showing how all of these things that we think of as intrinsically our own, what's the word I'm looking for? Free will, right? Like I'm in charge of my behavior, so I get, but really there's so many of these other factors that you don't even really kind of consider or think about that sort of bump up against all of these other things. And it's just part of living in a group and living exposed to different viruses and pathogens and things like this. So to me, it's like on a big picture level, that's kind of what's really fascinating. I want to throw one more at you, and we could start to wind down now. We've almost an hour, so don't take too much of your time. But what is it about ants that they seem to be the ones that get infected with all the crazy, host manipulating ones? And I think in some ways, some of them, like you can look at cordyceps, right? Cordyceps fungus, which everybody, I think, that's the one that most people know about, right? The fungus that grows in the brain and comes out and that kind of thing, right? That makes sense because it's the fungus, the way it disperses, it wants to release its spores when there's a lot of individuals around. So you have high density of social insects. That makes sense, right? Like that makes sense from the parasite point of view. The one that you're studying that may or may not be a manipulation, the transmission could be from, being close to each other. That's maybe not totally known yet. But the one that I looked at, dicrocelium, the transmission of the parasite is not relevant on being close to other ants, right? The ants get the parasite by eating this excretion from a snail, from the first host, the snail. And then once they're infected, they don't pass it on to other ants. They're just an intermediate step. But yet again, evolution has allowed this parasite to really, really finely control this ant. I think it's one of the examples where you can really, it's met the high bar of parasite manipulation, But it's just, there's something about ants, it seems, that they're the ones that seem to get hijacked by these things all the time.
And of course, you have a handful of those examples, and it's hard to say whether that represents really, how... It's hard to say whether ants are really kind of overrepresented in those cases. But I think it's easy to imagine why they might be. As I said, they are everywhere in very large numbers. And in a way, from a parasite perspective, they might... they're just kind of numerous. Like you can find them everywhere.
They're an easy target in that sense,
Exactly. And then, of course, not in the case of dicrocelium, but in the case of directly transmitted or parasites that only live in ants or social insects and that transmit from ant to ant, for example, a colony is like ideal, right? You never have, like a social insect colony is perfect. Like you have a very high number, very high density of hosts. They live in those, protected colonies that have ample resources that are homeostatic, as we said. Often they have stored resources. The temperature is stable, and so on. And all those hosts are not only plentiful, but they are also generally genetically related. So if you can infect one, the likelihood that you can infect all of them is very high.
Right, yeah.
And so that's this idea that from a parasite perspective, social insect colonies are extremely attractive, should be very attractive, right? And so again, that's an appealing idea. It's hard to really test because again, like the ants are pretty good at defending against that. So everything you're ever observing is just one snapshot of an arms race. But I think it makes sense.
Yeah. Yeah, it does make sense. And again, I think just to sort of maybe start summarizing, sort of the takeaways of this conversation and of this work, to me, it comes back again to this, the cost and benefit of group living, right? So you have this extreme example of group living in social insects where maximum benefit, they found a way to maximally benefit social living. And as you said, taken over a large section niche of ecosystems all around the world. So it's clearly very successful. But with every lifestyle, there's vulnerabilities, right? That pop up from that.
Exactly.
You can never plug all the holes, right? So finding these balances, and I just maybe you don't have to speculate too much out of your wheelhouse if you're not comfortable, but is there something that we can say then about this work, about this sort of relationship between behavior, infection, group living, that obviously ant to human comparisons aren't totally necessary, and we're not asking for public health advice, but what is sort of a big takeaway from an evolutionary perspective or something about the dynamics of group living, infection, that we can maybe start to take away from this kind of conversation?
Yeah, that's a that's a very interesting question. Not an easy one, I would say. So I think, yeah, definitely, I think it's pretty clear that social life has been very successful, that it works for ants really well. Humans are very social and very clearly dominate the planet by now. And I think so, like, of course, there are risks associated with that. You can have conflict within a group, and disease is an obvious one, and it's very fresh on everybody's mind because we have been faced with a very recent event of a major infectious disease outbreak. But also, I think one major takeaway is that both for ants and humans, societies are not powerless when it comes to fighting disease, and that sociality itself is allows to evolve defenses that are really effective as well, and that you could not deploy in a solitary context, right? So all this social care, right? Help, taking care of your sick nestmates in the case of ants, or deploying those collective defenses in the terms of how you organize society and so on. That's also very effective. And ants actually across the board very good, I think, at fighting pathogens and parasites. And you could argue that humans also are overall. I think we're doing pretty well, right? And then there's also, I think, communication is also a very important factor in that. So, for example, the speed at which we can exchange information or detect cases, early detection of infection cases is a big, I think, advantage that you have in a social context. So ants are also very, very good at detecting sick nestmates or detecting, the presence of pathogens in the nest. And I think that's also one of the big advantages that humans have at their disposal, right? Communication, even though it's very susceptible to, of course, also misinformation.
Yeah, fascinating. Actually, it's a good point, and maybe I kind of wish we would have gotten there a little earlier, but I didn't think of it. But yeah, it's like the benefits of social living. There's an exposure, but the sociality actually, has unique defenses as well that, like you said, you couldn't get if you're living on your own. So that's really, I guess that's the takeaway, is like social living has all of these advantages. And one of those advantages is also dealing with infections. That's ways to deal with infections. That's really cool. This has been a really, really great chat for me. Thank you so much for taking the time. I look forward to seeing the results of the ant fight experiments. So I'll be looking for those when they come out and learning more about this nematode. So I'll be sure to be watching, and maybe when you have some more results, you could come back and we could discuss those too. So thank you so much for taking the time. I really appreciate it.
Thanks for having me. This was really fun.
And thank you all for tuning in. I hope you enjoyed that conversation and were maybe reminded that, hey, working together is actually a pretty good thing. As always, again, rate, review, subscribe. Please, please, please. That is the best way to help out the show. Follow us on Instagram and Twitter at 2bradforyou. Subscribe wherever you're getting your podcasts. Hit us with an email at 2bradforyou@gmail.com. DM us, all of those things. We want to hear from you. We are available on podcasts for YouTube, so you can check us out there if you're using that. And maybe we'll expand into video. Let's see. Maybe I just need a push from you. So reach out. Let me know what you want to see, what you want to hear, what you want to talk about. Thank you, as always, to the Freak Motif for the music. That's all I have for now. Take care, everyone. We'll talk to you next time. Bye for now.